Hi guys, you're watching my YouTube channel BioHub and I'm your facilitator Dr. Seema Sharma. Friends, in this video you will get to know of verification of conditions necessary for photosynthesis. Photosynthesis is the process by which green plants manufacture their food. This process can be represented with the help of this equation. And these are the 4 conditions which are necessary for the process of photosynthesis. And here we are going to verify that carbon dioxide is necessary for photosynthesis. The aim of this activity is to show that CO2 is necessary for photosynthesis. To carry out this activity, we need potted plant, conical flask, split cork cap, stand and potassium hydroxide. We also need beaker containing water, test tube with holder, Bunsen burner, wire gauge, tripod stand, ethanol solution, petri dish, iodine solution, forceps and lighter. So now coming to the steps to carry out this activity. Step number 1, take a plant with de-starched leaves. You know viewers, during photosynthesis, glucose is formed. This glucose, it is converted into starch in the leaves and then it is transported to each and every part of the plant. When we are keeping our plant in some dark area, then the chances of photosynthesis will be very low. During that time, the stored starch, which was meant to transport to different parts, it will be reconverted into glucose and it will be utilized by the plant for its basic requirements. So after 2 days, these leaves, they will not have any starch in them. So this is de-starching. Now you know what is the meaning of plant with the de-starched leaves. In step 2, we have to insert one of its leaf in a conical flask containing potassium hydroxide. This is very important and very interesting chemical. It has the ability to absorb carbon dioxide gas. Step 3, we have to leave our experimental setup in bright sunlight for 5 to 6 hours. So this is our experimental setup, friends. This is an airtight experimental setup. We are going to perform this experiment on 2 leaves, one which is inserted in the conical flask labeled as leaf A and another which labeled it as leaf B. So after 5 to 6 hours, in step 4, both these leaves, they are subjected for starch test. First, we have to keep the leaf in the boiling water for 5 minutes. Then we have to put these softened leaves gently in 2 separate test tubes containing ethanol. We have to keep these test tubes in water bath for 7 to 8 minutes. And here you have to remember that this step should be carried out very carefully because ethanol can catch fire very easily. Next, we have to take out and rinse these leaves with tap water and we have to place these on petri dishes. Then, iodine will be poured on these and we have to note the reaction. That is, we have to note the change in color. Iodine is a universal indicator of starch. In whatever material starch is present, if we are pouring iodine on that, immediately it will turn blue-black. So what is the expected result of starch test? So observation with this experimental setup is the leaf which was inserted in conical flask, leaf A, it has shown no reaction with iodine solution. On the other hand, leaf B turned bluish black in color when tested with iodine for starch. With the help of these 2 observations, we can conclude that the KOH in the flask, it absorbs all the carbon dioxide. Thus, leaf fails to carry out photosynthesis to make glucose and then form starch from that glucose. So, with the help of this activity, we can clearly conclude that carbon dioxide is necessary for photosynthesis. Now, let's switch over to the precautions. Precaution number 1, before carrying out the experiment, plant should be de-starched properly. Second, the glassware should be cleaned and wiped before and after the use. Third, the experimental setup should be completely airtight. Fourth, boiling of leaf in ethanol should be done in the water bath because ethanol is inflammable. Fifth, wear a lab coat while performing this experiment in your science lab. With this, it's time to sign out. Thank you so much for watching and supporting BioHub. Keep experimenting and keep learning.